In this video, we're going to explore technology called biotechnology. So we're going to take everything that we learned in genetics and take a look at these lab methods that are used to manipulate DNA and also analyze it. So we can actually now go in, we're at this point where we can go into an organism's DNA and manipulate it and modify it in some way to get some kind of result. So we've come to such an understanding of DNA and genetics that we can actually start to manipulate and, let's say, play with it. And there's so many different applications that can result from this type of, let's say, power or knowledge. The examples of biotechnology that we're going to study in this video are GMOs, gene therapy, gene cloning, something called DNA fingerprinting, just briefly, reproductive technologies, which you've already seen, and then genetic testing, which we've seen as well. I won't go into too much details on the stuff that we've seen. I'm just going to give you a general overview of each, and you're going to read more about them, and even do some of your own research, learn about the current trends in biotechnology today. And this might be something you're interested in studying in your future. You might want to do a master's on this or a PhD on this and work with this throughout your undergrad, because it's quite an interesting field, a developing field of research. So before you begin, one thing I want you to do is watch this video over here. And what I want you to get from this video is that people often form an opinion about biotechnology without really knowing anything about it. So for example, if I had asked you what GMO meant or what it means, or let's say I started off by asking you, do you agree with GMOs? Would you have an opinion before you actually research the facts? When you watch this video, you'll find it quite humorous to see that a lot of people do have this opinion about GMOs without even knowing what GMO stands for. So a GMO is a genetically modified organism, hence GMO, meaning that it's an organism that was created through some kind of genetic DNA modification. So, as I mentioned, the definition is an organism whose genetic material has been modified using genetic engineering techniques or tools. So you modify the DNA of an organism to get some kind of characteristic that you're interested in. One interesting type of GMO, genetically modified organism, is called a transgenic GMO. Trans meaning across. So transgenic means you take genes from a different species and put it into another species. So if you take a look at this example here, we have a zebrafish. And what scientists have done is they've actually taken a gene from, let's say, a jellyfish that's able to fluoresce, that's able to give off light, and they put it into this this regular zebrafish here. And when they put that gene into the zebrafish, now the zebrafish is able to also produce the protein that allow it to fluoresce and give off light when it's exposed to different types of light like UV or some blue light. And so this is an example of a transgenic GMO, a fluorescent zebrafish. In stores, you can actually buy them, and they're called glowfish. Now, they were illegal in Canada, but I actually found out this weekend that they're now being sold at PetSmart in Canada, and I walked in just to see if that was the case, and they are. On Saturday, some of the PetSmarts are holding an educational seminar to explain how glowfish work and how to take care of them and how to buy them. They're really, really cool, and they come in all these different colors. So the green ones, they have a gene from a jellyfish, and the red ones have a gene from a different organism that allow them to fluoresce or to give off light of the red color. So they're really, really neat, and they look really, really cool. Much cooler than the, let's say, boring one that you see over here, this boring color over here. So I'm going to go over in general how genetically modified organisms are made. Before we do that, though, the glowfish didn't seem like a very practical thing, so I'm going to show you another great example that's a bit more practical. One example of a transgenic organism is the antifreeze crops. So they're essentially plants, so plants that grow things that produce things that we eat, like fruits and vegetables. There's genes from a fish put into the plants, and these genes are called antifreeze genes. They basically produce protein that allow the plants, the fruits, the vegetables to survive in colder temperatures and to survive transportation when it's cold, and so things can last a little bit longer in those cold storage areas. So you can see the process over here. We have a flounder fish. We find the gene that we want to put into a plant from the flounder fish, from one of its cells here. We cut the gene out and we put it into this round bit of DNA over here called a plasmid. So this plasmid is just a round bit of DNA. It acts like a vehicle to carry the antifreeze gene, or whatever the gene is. It doesn't really matter. It could be the glowing gene for the zebrafish. But the main idea is that we put this antifreeze gene into the plasmid, and then we put the plasmid into a bacterial cell. And what happens is this bacterial cell, now that it has a plasmid, is going to reproduce like crazy and make many copies of this antifreeze gene in here. The other thing that's going to happen is eventually what we're going to do is we're going to take the plant cells and the DNA made from the bacteria, combine them together, so now the plant cells will contain the gene of interest, the antifreeze gene, and then once those plant cells contain the antifreeze gene, they can grow and become plants that are tolerant to that cold temperature, and the products of that plant will survive colder temperatures, and it'll be better for the economy, and they'll survive again those longer transportation routes. So the idea is that we took a gene from one organism, the flounder, and then eventually put it into another organism, this tomato plant over here. So there's antifreeze tomatoes, antifreeze strawberries, and it's all because of that gene that was put into them. So the main idea is that we take a gene that we're interested in from an organism, cut it out, put it into a plasmid. The plasmid is just a circular piece of DNA that can carry our gene that we're interested in. We put that plasmid into a bacterial cell and allow it to reproduce like crazy, and then we inject that plasmid DNA with the gene of interest, the antifreeze gene in this case, into the plant, and then the plant will now have that gene, and it can make proteins that allow it to have those antifreeze properties. And that there is called a transgenic organism now, because it has genes from the plant has genes from a different organism, from a different species, from the flounder in this case. So as you can see, there are some practical applications as well to making genetically modified organisms. It's not just to make the cool glowfish. So this is a more detailed explanation of how the transgenic organism is made. You can see over here we have the Antarctic cod, which is the fish that has that antifreeze gene. We decide to cut the gene out using something called restriction enzymes. Restriction enzymes are special types of proteins. We often call them DNA scissors. They cut DNA in very specific regions. As long as we know the general nucleotide sequence of that gene we're looking for, we can cut it out specifically. We use a restriction enzyme on the plasmid as well, so we can cut the plasmid open. The plasmid comes from a bacterial cell, typically. It's how you find DNA in bacteria. In organisms like us, like humans, our chromosomes, our DNA is found on linear chromosomes like this, but in bacterial cells it's found on those round plasmids. So we use restriction enzymes to cut the plasmid, and then we can take our gene of interest and put it into the plasmid. To glue the gene of interest into the plasmid, we use something called essentially DNA glue. It's DNA ligase. It's an enzyme that connects different pieces of DNA together. And so at this point now we have recombinant DNA, which basically means DNA made from two different sources. We then take that plasmid and then put it into the bacterial cell, and the bacterial cell reproduces like crazy really, really quickly, and it reproduces everything about itself, including that antifreeze gene. We then take the bacterial cells, put them into the same environment as the plants. The plasmid will go from the bacterial cell to the plant cell, and our plant cell will have that new gene in its genome, and genome just means a collection of DNA. And so now we have that transgenic plant that's made. And so this is in general how those transgenic organisms are made. It might be a good idea to know the process of making a transgenic organism and knowing a few examples of them. You might want to research some on your own, some that you find interesting, and bring them into the classroom for discussion purposes. Another really cool one is something called Bt corn. Bt corn has a gene put into it from a bacterial cell called Bt bacteria, and that gene allows the plant to basically produce its own pesticide. So here we can see the DNA that was cut out, the Bt gene. It was put into a plasmid, and then eventually it was put into bacteria to reproduce, and then eventually the gene of interest was placed into the corn cells, and those corn cells became corn, plants and corn, which now have the ability to produce their own pesticides and kill anything that tries to eat it. So again, another great application of creating transgenic organisms. But I'm sure, as you noticed, there's probably some negative effects to this one here particularly. That's right, these insects are dying. Yes, they're not eating the plants, but how controlled is this? Are we going to go too far and kill this insect population? Those are questions you need to ask yourself when you're doing science. Just because we can do something doesn't mean we should. And just because we can do something, or just because we are doing something, doesn't mean that we should let it run wild. Should we put in some limitations? Should there be some laws that regulate this? And there are. And that's a good thing, and that's something that you could study as well if you decide to go more into biology in the future. Another really cool example of transgenic organism is golden rice. Golden rice has genes added to it, two genes actually, that allow the rice to produce more vitamin A. And the reason we're doing that is because in African countries, many children were going blind because they had vitamin A deficiency. There just wasn't enough vitamin A in their food. But now that this golden rice has been created, the rice that these people eat has a lot more vitamin A, and it prevents that vitamin A deficiency blindness. So again, another great application of GMOs, right? So when people say GMOs are bad, it's messing with nature, there's no benefits to it, you need to really question what they're saying. You need to question them, ask them, do you even know what a GMO is? Do you even know how it's made? Do you know that there are many benefits to GMOs? Yes, there are questions and ethics behind every scientific practice that you do, but you need to educate yourself about what the practice is first before you can actually judge it truly and totally knock it. Here's another great application. So if you suffer from diabetes, you might need to take insulin injection. And so when you buy insulin, well, you might often wonder, where is it coming from? So you can see here, there's different ways you can do it, but we took an insulin gene over here, we took a plasmid over here, put the insulin gene into the plasmid, put the plasmid into a bacterial cell, and that bacterial cell reproduced like crazy, and now that bacterial cell has the insulin gene, and it's making some insulin, and then we can take that and basically produce it, market it, and give it to people sell it to people who have diabetes and need that insulin. So again, we're mass producing insulin using this technique of genetically modified organisms. So this bacterial cell is a genetically modified organism. It's a recombinant bacterium now. It has DNA from different sources, different species, not just its own. Another great example, and there's actually many more examples you can read up on page 247 of your textbook. You should read up on spider silk to see how it's made, what the applications are. Another great application of biotechnology is something called gene therapy. Now this is still in its baby steps. It's not super advanced yet. There are definitely some side effects. Things don't always work the way we want them to. We're not super specific with this technique yet, but it seems very promising so far. So the idea behind gene therapy is that, let's say there's a disease that's a genetic disease, and you know where that defect is for your gene. You can go and cut out the defect and then replace it with a good copy of the gene. We often use it in cancer treatments, but there's other examples that we'll talk about in just a bit. But the idea is that we use a virus. So a virus is all a virus really is. You've probably heard of the term virus before, but you don't really know what it is right away. It's a case, a protein case with some kind of genetic material inside of it. Viruses don't really follow all the characteristics of living things. For now, I just need you to know a virus is a protein case with some kind of genetic material in it. And so what we do is we can take the, let's say there's a sick person here. We can take cells from the sick person. So these are some cells taken from the sick person. We can then go and get a virus, and what we can do is we can get a good copy of the gene and put it into the virus. So this person's cells has a bad copy of the gene. This virus, we put a good copy of the gene into it. We then put that virus into the person's cells, and then that virus with the good copy of the DNA will insert the good copy of the DNA into the person's DNA. And so the rest of that person's cells will now have the good copy of the DNA, and hopefully the symptoms of the disease will decrease or altogether disappear. And now that's assuming that we're very specific. If we can be specific by finding the exact location to cut these in, that would be ideal. We're not really 100% there yet, but we are developing some interesting technology that's allowing us to get there. So here's some more detail on how gene therapy works. Here we have our target cell. So this is our cell that has the bad gene, the faulty gene that we want to fix. The vector in this case is going to be a virus. So in the transgenic organisms that we saw earlier, we were using a plasmid and bacterial cells. Here we're using a virus as a vector, the vehicle to carry our good gene in. So we put a good gene into the virus. That virus will then enter the target cell. It will make its way to the nucleus where the DNA is kept. It'll inject the good gene into the nucleus, and then that gene will hopefully become part of our genome, our DNA collection, and now the gene will no longer be faulty. We'll hopefully have healthy cells from now on. And so gene therapy does have some successes. It's often used to treat Leber's congenital amaurosis, which is essentially a disease that affects photoreceptors in your cells, so in your eye cells. And those photoreceptors are essentially the cells that pick up signals from light and then help to control your vision. So we actually do use gene therapy to treat this condition. I do want you to read up on it in some detail, so you can see it on page 251. And then I also want you to read up on the drawbacks and obstacles that are in the way of gene therapy. So maybe list a few of the drawbacks, a few of the obstacles. It's important to understand that there are successes, but also important to understand that there are drawbacks, so we can learn from those drawbacks and hopefully move forward and improve the technology. Another example of biotechnology would just be the reproductive technologies that we've discussed already. The artificial insemination, where you put the sperm into the uterus of the female, you inject that there. The in vitro fertilization, in vitro means glass, where you prepare the organism in, let's say, a petri dish. You prepare the zygote there, and then you go and you implant the zygote into the uterus, into the mother, and then hopefully it'll develop there. So this is another example of biotechnology at its finest. It's helping couples that can't conceive actually conceive children. One interesting tidbit of information is about this woman here named Louise Brown. She's actually the first test tube baby. She was born in 1978, and it was kind of a miracle that you're able now to produce these babies outside of the womb, and now you're giving couples who couldn't produce children before the ability to have kids now. So yes, there are ethical issues with many of these biotechnological techniques, but there are many benefits as well that often make it worth exploring. It's all about analyzing the cost versus the benefit. If the benefit outweighs the cost, if the good outweighs the bad, maybe it's something that's worth exploring. Some more biotechnology includes genetic testing, where you can grab DNA from the developing embryo or surrounding cells, and then analyze it, create a karyotype to see if there's any chromosomal abnormalities or genetic defects. And we've already discussed this in previous presentations. It's always good for you to review this to know that practical application. One downfall of the amniocentesis is when you collect the amniotic fluid, it can increase the risk of a miscarriage and infection. So it's always something, if people go this route, they need to have the proper education and facts before they make that decision. So it's always about making that informed decision. And another very interesting direction that people are thinking about now is this idea of a designer baby. If we can go in and manipulate DNA, cut pieces of genes out, put in genes that we want, we're getting to this point where maybe one day you'll be able to completely get rid of a certain disease. You'll be able to maybe design your baby, choose its eye color, try to choose its sex, try to choose how tall it might potentially become. And that's what people are kind of afraid of. How far are we going to go with this biotechnology? Are we going to start marketing it where you can go and design the baby that you want? So what I want you to do is watch the next series of videos here. It's on a new, relatively new piece of biotechnology called CRISPR. And you'll see it's quite the amazing tool where we have isolated this, let's call it biological toolbox, enzymes and glue, and we can go in and precisely cut out DNA with precision that we've rarely seen before. And we can put in genes in areas with extreme precision. And this has actually been used as of late. So I do want you to read up on it. I want you to watch these videos so you can learn about what the CRISPR technique is and what the benefits are and also what the downfalls of it are. What are the ethical dilemmas behind it? So do watch these videos, do understand the general idea behind it, because you're going to be hearing a lot about this in the news. Whether you're a biology student or someone who tries to avoid science for the rest of their life, this is going to be in the news. This is going to be a topic of conversation. This is something that you want to read up on and learn about. So at least you're informed about it and you're not like those people in the video at the beginning of this presentation for GMOs. So the idea behind this is that one day we'll be able to design our own chromosomes. This is a simplified idea, of course, but people are afraid that if we can design our own babies, that we are going to start being we're going to start discriminating at the genetic level. And so a really neat movie that hopefully we'll have time to watch this year, but if we don't, is called Gattaca. And it explores the whole idea of essentially using genes to determine where you will fall in society. And so if you're quick, you may have noticed the title Gattaca. Think about how it relates to genetics. What are those letters? What do they mean? What is this particular stretch of letters? What does that mean? So if we do have time, we'll watch this movie and we'll do a little analysis on it and discussion. If not, it's definitely a movie that you should put on your to-watch list. It's one of those movies that when you start off watching it, you may not like it immediately, but you get hooked. It's a really good movie, one of my favorites. So in order to learn about this concept in more detail, the associated readings can be found on page 245 to 251. Do pay attention to the examples of biotechnology as well as the benefits and the downfalls. So make a list of those. There are some great practice problems for you to attempt. Questions 1 to 6 on page 248. Questions 1 to 3 on page 251. And then also the learning goals that are posted on the course website. So please make sure you attempt those. Make sure you understand what biotechnology is, just basically using principles of science in order to manipulate and to analyze DNA. We saw transgenic organisms, which are a type of GMO. We saw gene therapy. We saw reproductive technologies. And I also highly encourage you to watch the videos on CRISPR technology so you understand the new direction in biotechnology today. Here you can see the learning goals that are related to this PowerPoint and the readings from your textbook.